Hello everyone. Welcome to today's webcast, Expanding the Limits of Shotgun Proteomics Using Micropillar Array Columns and Data-Independent Acquisition Methods. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this web seminar presented by LCGC and sponsored by Pharmafluidics and Biognosys. Pharmafluidics is a spin-off of the Chemical Engineering Department at the Free University of Brussels under Professors Geert Desmet and Wim de Malsche. Pharmafluidics is bringing to market a game-changing technology in analytical chromatography for biomarker discovery and the analytical development of biopharmaceuticals. Their micropack columns are made by etching and have a separation bed of highly ordered and freestanding pillars. Due to this unique design, the micropack columns have the capacity to offer high separation power even for tiny biological samples with an excellent level of resolution, unprecedented robustness, and unrivaled reproducibility. Biognosys AG is a leader in next-generation proteomics, dedicated to transforming life sciences research by making the most advanced proteomics tools available to researchers. Biognosys offers a suite of products and services to decode the proteome and equip researchers from all fields with a deep understanding of protein expression and quantification in a biological system. We have a few housekeeping announcements before we get started today. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of your window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the webcast. And if you have any technical problems today, viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speakers. We are very pleased to be joined today by Dr. Jeff Op de Beeck and Dr. Jan Muntel. Jeff Op de Beeck was Pharmafluidics' first full-time employee. His PhD work under the supervision of Professors Geert Desmet and Wim de Malsche formed the basis for the current micropillar array column, which is branded by Pharmafluidics as the micropack. He holds a master's degree in biomedical sciences from the University of Antwerp and a PhD in chemical engineering from the University of Brussels. He currently holds the position of Application Development Manager at Pharmafluidics. Jan Muntel has been employed as a senior scientist on the R&D team of Biognosys AG since 2016. He has long-standing experience in quantitative proteomics. His work at Biognosys is focused on the development and benchmarking of new LC-MS workflows. He holds a PhD from the University of Greifswald and was a postdoctoral researcher for 4 years at Harvard Medical School. Thank you both for joining us today. Jeff, please go ahead and get us started.
Thank you, Laura, for the kind introduction, and thanks again for giving us the opportunity to set up this webinar to present and explain our innovative approach to making nano-LC columns, and more specific about the benefits they can bring to single-shot proteomics using data-independent acquisition methods, which will be covered by Dr. Jan Muntel from Biognosys. I also want to welcome and thank the participants for their time and interest in our product and technology. This is what we will be covering today. First of all, I will give an overview of the micropillar array column technology and the analytical performance of the products we are offering. Then Jan will take over and start with an introduction to data-independent acquisition methodology, after which he will cover the benchmark of data-independent acquisition, the optimization for micropillar array columns, and comprehensive single-shot analysis of HeLa samples into more detail. Finally, we will both be available for answering any questions you may have. At Pharmafluidics, we use a fundamentally different approach to make chromatography columns. This allows introducing perfect order into the separation bed. Now let's first look at the design of traditional LC columns. The stationary phase of traditional columns consists of a bed of microparticles that have been packed into a cylinder or capillary tube. These particles have a certain size distribution and thus a degree of heterogeneity. On top of that, their location in the column is more or less random, at best not perfectly ordered. As illustrated in the bottom figure, this disorder will lead to additional dispersion as the sample migrates through the column. The fact that not all flow paths in the column are identical will cause sample molecules to have slightly different retention times, causing additional dispersion. Pharmafluidics uses a different approach to fabricate their chromatographic beds or columns. Instead of using particles that are packed into a tube or capillary, the backbone of the stationary phase is designed in a similar way that electronic circuits or sensors are designed. State-of-the-art lithographic techniques followed by silicon etching are used. These channels are filled with accurately positioned ordered arrays of pillars which form the backbone of the stationary phase. This results in two important benefits. First of all, virtually perfect order is achieved, hereby reducing the eddy dispersion to an absolute minimum. This is again illustrated in the bottom figure, where you can see that all sample molecules will follow identical flow paths throughout the entire column. An additional benefit of this format comes from the fact that the pillars are freestanding and positioned at an accurately defined distance from each other. This has a serious impact on the column permeability and enables us to provide very long columns that can be operated at moderate pressures. The benefit of order versus disorder is clearly illustrated in these animated simulations that have been generated in Professor Geert de Smet's group and that formed the basis for the current micropillar array column. In the disordered case, the sample plug gradually disperses as it migrates through the stationary phase, resulting in significant broadening during the separation. Whereas in the ordered case, the sample plug remains nicely conserved and the intensity remains high, resulting in improved resolution and higher signal-to-noise ratios. Now, to make long columns on limited footprint, the columns need to be folded. The current production process is based on 4-inch silicon wafers, one of which is shown in the middle picture. And on such a wafer, we realize 3 2-meter long columns and 2 50-centimeter long columns. A 2-meter column actually consists of 40 discrete lanes that are interconnected by 180-degree turns, finally resulting in a 2-meter long meandering or serpentine channel. Making microfluidic turns without introducing additional dispersion is quite difficult because the racetrack effects where the inner lane is shorter than the outer one need to be avoided. To avoid these effects, we use proprietary flow distribution and collection structures at the beginning and at the end of each lane. Then, at the bottom of this slide, we gradually zoom in upon the pillar surface from right to left. Once the pillar structures have been etched, they are rendered superficially porous to increase the effective interaction surface. The final result are channels filled with 5 micrometer diameter pillars, positioned at a distance of 2.5 micrometer. The height of each pillar is 20 micrometer and the cross-section of the lanes is equivalent to a circular capillary with a diameter of about 85 micrometer, or 58 micrometer. The porous layer is around 300 nanometers thick and the average pore diameter is estimated at about 10 to 30 nanometers. The bonded phase for the current products is C18. A feature that was very helpful during the research and development phase is that our columns have a transparent glass cover lid, allowing us to follow what happens inside the column using fluorescence imaging systems and fluorescent dyes. The first video shows a real-time injection of a fluorescent dye into the column. It illustrates nicely how the injection is controlled by the flow distributor in order to uniformly distribute the sample over the relatively wide separation bed, and how the separation bands remain nicely straight and parallel. The second video shows how the proprietary collection and distribution structures allow to conserve the sample plugs in a 180-degree turn. What we see here is not a real-time separation, but instead a number of sequential low-volume injections. Combining these two principal capabilities, we can provide very long columns that maintain sharp peaks. Now let's have a look at the performance that can be achieved by using these columns. In this slide, we compared a 50 centimeter long micropack column to 2 state-of-the-art commercially available nano LC columns, which are widely used among proteomic labs. As our 50 centimeter column is ideally suited to perform relatively short analysis, we compared it to 15 centimeter long pack beds alternatives. The sample used was a HeLa digest and it was separated using short gradients ranging from 30 to 90 minutes. As can be seen from the top 2 chromatograms, The micropack column was operated at 2 different flow rates, whereas the packed bed columns were only operated at a flow rate of 300 nanoliters per minute. The top bar chart displays the average peak width that could be obtained for a retention time calibration mixture that was spiked into the HeLa sample. Compared to both packed bed columns, a clear reduction of the peak width could be achieved. What also stands out is the pump pressure needed to operate these columns. As you can see in the bottom chart, the pressure needed to operate a 50 cm micropack column at a flow rate of 300 nanoliters per minute is only 40 bar, whereas both packed bed columns delivered column back pressures exceeding 250 bar for this flow rate. Because of the low back pressure, 50 cm long micropack columns can be operated over a wide range of flow rates, going from 0.1 to 2 microliter per minute. In this case, an example of operation at 1 microliter per minute is given. As can be seen from the charts shown on this slide, the increase in chromatographic performance also translates into an increase in peptide and protein identifications, especially for short gradient times with a total analysis time within 60 minutes, where more than double the amount of peptides and 72% more proteins could be identified when operating the 50 cm micropack column at a flow rate of 1 microliter per minute. In another set of experiments, we benchmarked our long column, the 200 cm long micropack column, towards a longer 40 cm packed bed column. On this slide, base peak chromatograms obtained for the separation of a human embryonic kidney cell sample have been compared. The top one, displayed in orange, was obtained on the 40 cm packed bed column, whereas the bottom one, displayed in blue, was obtained on the micropack column. A quick inspection of the chromatograms already suggests that more features would be detected in the top chromatogram. These assumptions were confirmed once data had been processed, with a total of 3,034 protein groups identified with the micropack column compared to 2,334 protein groups with the 40 cm packed bed column. Both columns were operated at a flow rate of 300 nanoliters per minute and at a temperature of 50 degrees Celsius. For the micropack column, this resulted in an average column back pressure of 75 bars, and for the packed bed column, this resulted in a back pressure of 445 bars. Now let's have a look at the chromatographic performance for these long columns. The peptide peak widths obtained for the separation of these samples have been plotted as a function of the gradient time. For short solvent gradients, similar peak widths were obtained for both columns, but when extending the gradient time beyond 2 hours, there is a clear difference between both column types. This becomes even more obvious when the average peak width is plotted. The rate at which the peak width increases according to the gradient time is much lower for the micropack column as compared to the packed bed column. But how does this translate again into peptide and protein identifications? A 30 to 50% in peptide IDs could be achieved over the entire range of injections, with the lowest amount of 100 nanograms shown to the left and the highest amount of 3 micrograms shown to the right. On the protein level, this resulted in a consistent increase in identifications of 20 to 30%. Extending gradient times beyond 4 hours definitely has a positive effect when working with a micropack column, ultimately delivering over 5,000 protein and 40,000 peptide identifications for a long gradient ran on an Orbitrap Elite instrument, and this in data-dependent acquisition mode. With a column volume of 3 microliter and a flow rate range from 0.1 to 2 microliter per minute, the 50 cm micropack column is ideally suited to perform more routine analysis where increased throughput is needed. To serve those who are aiming at comprehensive proteome coverage, Pharmafluidics offers the 200 cm long micropack column with a volume of 9 microliter and a respective flow rate range of 0.1 to 1 microliter per minute. What is also quite important is the configuration needed to connect the column properly to ESI-MS instrumentation. Because the column is mainly made out of silicon, which is a semiconductor, a dramatic impact on the chromatographic behavior of micropack has been observed if no proper precautions are taken to shunt the high voltage to ground. The schematic drawings on this slide clearly show how the column should be connected, and this for 3 different electrospray ionization configurations. The column is provided with 2 stainless steel unions. On the inlet side, standard 360 micrometer outer diameter tubing can be connected without introducing any dead volume. And at the exit side, a union with a 50 micrometer through bore is provided to properly ground the column and protect it from the high voltages applied for electrospray ionization. For those who are working with a thermoscientific easy spray ionization source, we also offer a micropack compatible easy spray emitter that can easily be connected to the micropack column and fitting. This closes my part on the micropack column technology. I'm very pleased now to hand over the floor to Dr. Jan Muntel, who will share his experience with our micropack columns in combination with data independent acquisition methodology. But I think it's first time for some polling questions. Is that correct, Laura?
Yes, that's correct. Thank you very much, Jeff. I would indeed like to ask all of you in the audience to participate in 2 very brief polling questions. And the questions are on the first question's on your screen, and you can click directly on your screen to enter your answers. So here's the first question. What do you currently find to be the most challenging aspect of applying nano-LC methods for deep proteomic analysis? Maximizing LC resolution, limited sample availability, maximizing the number of identifications, maximizing the plate number, reducing the runtime, maximizing reproducibility, or increasing column stability. So once again, you can click directly on your screen to enter your answers. And once again, here's that question. What do you currently find to be the most challenging aspect of applying nano-LC-MS for deep proteomic analysis? Maximizing LC resolution, limited sample availability, maximizing the number of identifications, maximizing the plate number, reducing the runtime, maximizing reproducibility, or increasing column stability. All right, let's pull up those answers. Okay, so it looks like by far the number 1 answer is maximizing the number of identifications. Jeff, did you have any comment on those results?
Yeah, so I think maximizing the number of identification is indeed something that can be achieved by using our very long column, the 200 centimeter micropack column, and then running it with very long solvent gradient times. But I think also the data independent acquisition methodology of Biognosys will help to do so. So I think now we can go further to the part of Jan.
Excellent. Before we do that, we just have one more quick question for the audience, one more polling question. So that is now on your screen, and again, you can click directly on your screen to enter your answers. So here's that question. Which is your preferred quantitative proteomics workflow? Label-free workflow based on DDA methods, isotopic labeling workflow, SILAC, TMT, iTrack, SRM, MRM, or PRM-based quantification using labeled peptide standards, AQUA, label-free workflow based on DIA methods, or I'm not working in the field of proteomics. So once again, click directly on your screen to enter your answers. And that question is, which is your preferred quantitative proteomics workflow? Label-free workflow based on DDA methods, isotopic labeling workflows, SILAC, TMT, or iTrack, SRM, MRM, or PRM-based quantification using labeled peptide standards, AQUA, a label-free workflow based on DIA methods, or I'm not working in the field of proteomics. All right, let's pull up those answers. I'm not seeing the answers come up on my screen. So let's just move on. Jan, why don't you take over and do your part of the presentation.
Thank you very much for the introduction. So welcome to the second part of this webinar about expanding the limits of single-shot proteomics using micropillary columns and data-independent acquisition methods. At the beginning, I would like to briefly introduce our company. So we are typically working on discovery proteomics project. That can be biomarker discovery, drug target discovery, mode of action studies, as well as drug target deconvolution studies, or we also do studies where we look into post-translational modifications. For the kind of samples, we basically work with almost all types of body fluids, for example, plasma or urine, but we have also long-standing experience with tissue or cell line samples. These samples can be derived from a whole bunch of different organisms, as human, mouse, rats, but they also had some plants or bacterial proteome projects. Our experiments can be rather small, so just comprised of 6 samples, but we have also some projects where we look into hundreds of different samples. That can be time course experiments, treatments, and other studies. For all of these studies, we have some key requirements into our technology. One is we want to have a high proteome coverage that also goes along with high sensitivity. We want highly reproducible technology, high quantitative precision, and also the approach needs to be scalable. So to make sure that everyone is on the same page, so when I talk now about bottom-up or shotgun proteomics, the main challenge is that usually proteins are very large molecules and very heterogeneous in their physical-chemical properties, so they are not easily analyzable with one approach. Therefore, we digest proteins into peptides, basically cut them down into smaller units, and quantify later on the peptides as proxy for the proteins. The peptide properties are typically rather small, 7 to 25 amino acids, very homogeneous in the chemical properties, so they can be analyzed with the same analytical setup, usually stable and easy to handle. Typically in the field of proteomics, many people use, or in the past used data-dependent acquisition methods. So what's basically comprised is that after chromatographic separation of the peptides, MS1 service scan is acquired, in which an overview is generated of the peptides present in the sample. Then one peptide is selected and for fragmentation, and an MS2 spectra is acquired of it, which basically delivers the sequence information of these peptides. Typically, the peptides are chosen by the intensity in the MS1 spectrum. This can lead, as you can see in the small little map, to semi-stochastic sample of the sample. So that basically a lot of peptides cannot be analyzed because the scan speed of the mass spectrometer is not high enough. For that reason, we turned to use data-independent acquisition methods. The chromatographic separation is basically the same. Then the MS1 scan is acquired, and then the whole mass range is fragmented in different windows. This results basically in very complex MS2 spectra, but on the upside, we can achieve a comprehensive sampling of the whole sample. So people might ask, why is that important? So therefore, I'd like to show this picture. So here you can see a giraffe walking around. It looks like a very idyllic picture, but if you really increase the resolution and data points of this picture, you can actually see that the line shows up, so it actually comes down to a question of life and death for this giraffe. So as we can achieve comprehensive sampling and data-independent acquisition methods, a new challenge approaches, and that is to interpret the complex MS2 spectra. For that, we use the so-called peptide-centric analysis using a library. The library, also called ion library or spectral library, is a repository of previously acquired peptide sequences, fragment ions, including their intensities and basically retention times. In the first step of the analysis, the software tools detect the peaks in the MS2 spectra. Then extract and ion chromatograms are generated on MS1 and MS2 level. They are matched together and then scored. Then FDA estimation is done. And later on also, these intensities can be used for quantification of the peptides. All of these algorithms are actually implemented in our Spectronaut software. To benchmark the DIA method, we started off using a HeLa sample, Q-exactive HF mass spectrometer, and filtered the results on 1% FDR on peptide and protein level. What you can see here is the gray line, which is basically the number of theoretically possible acquired, or the number of spectrums that can be theoretically acquired by the mass spectrometer. So it increases linearly with increasing of the gradient length. The light blue line basically shows how many spectra are actually acquired by the mass spectrometer. So for short gradients, it's pretty close to what is theoretically possible. But with longer gradients, there's a bigger gap between that. And in dark blue or purple, that is the number of spectra that were actually identified in our lab with using these samples. And in orange, you can see what one of the best labs achieved in acquired. So still just around 30 to 70% of all spectra actually identified at the end. In contrast to that, in red, you can see how many peptides were identified with the DIA approach. It is within the range usually of double the number of identification compared to the DDA approach. And if you look at especially at short gradients, you can see that there are more peptides actually identified with DIA approach than are theoretically identifiable with the DDA method. With that, we were also very interesting how well it performs in terms of quantification. Therefore, we used a mixed proteome sample in which different proteomes are spiked into the sample in different predefined ratios. Used the same mass spectrometer for that. And the summary of the results you can see here. One is using the MS1 information of the DIA experiment. One using the MS2 information of the DIA experiment. And for DDA experiment, only MS1 information is available for label-free quantification. What you can see here is basically that for all predefined ratios, the DIA MS2 is closest to the predefined levels. And actually, meaning the accuracy is pretty high of the quantification. Also, if you look at the width of the box plot, they are also the smallest for the DIA MS2 approach, meaning that also the precision is pretty high. So with that, all results I showed you so far were acquired on a 50 centimeter in-house pack column with sub 2 micrometer silica particles. So in a standard experiment, we are able to quantify between 5,000 and 6,000 proteins with a 2-hour gradient. But for comprehensive coverage, a lot of people in the field say that around 10,000 proteins, and you achieved a comprehensive coverage. So we are still around just 50 to 60% of that, even though there are more than 60,000 NR transcripts described in mammalian cell. Still, the 10,000 is kind of the number to achieve. And for a single-shot proteome analysis at this point, we only have the chance to increase the gradient length. We also try to use longer gradient, but then longer columns, but then we quickly reach the upper pressure limit of the LC. So that is the plot that I showed before already. So with increasing gradient length, we are able to identify more and more peptides. But as you can see already, that with longer gradients, the increase in identification doesn't keep up with additional time spent on the separation. It becomes more obvious by plotting the IDs per minute. So the most efficient setup is basically 1 hour gradient, but after that, the precursor identification per minute are constantly dropping. And then 4 hour gradient, actually it's less than half compared to the 1 hour gradient. So why is that? So therefore, you can see here an overview about the full width and half max of the separation, which basically increase linearly with a longer gradient. And we also see a plot together the peak capacities that were achieved within this gradient. You can see by doubling the gradient from 30 to 60 minutes, we gain 100 in peak capacity. Doubling from 60 to 2 hours is 100 more, and doubling from 2 to 4 hours is again 100 more. Next slide, please. So then the limitation becomes more obvious if you plot the identifications per peak capacity. So what you can see, it's basically constant. So that means actually if we are able to increase the peak capacity for this LC setup, we are also able to increase the proteomic coverage. That is actually why we decided to try out the micropack column. So we started off with a 200 centimeter micropack column, which is the most promising column for high proteome coverage. We operated it at 300 nanometer per minute and heated up to 50 degrees. It is primarily to be able to equilibrate and load the samples at higher flow rates to increase the efficiency of the setup a bit. We use a thermo-easy nano-LC, a thermo nanospray flex ion source using stainless steel emitters, and a QX-active HFX mass spectrometer. That is how it looks like in our laboratory. So within the beige brown bag, you can see the column, so it's actually hidden inside the heater, and it's connected to our HFX mass spectrometer, which listens to the name of groundhog. Therefore, you can see the small little animals on it. Before we actually looked into the performance of the column, we started to optimize the gradients. For that, we started off with a linear gradient and calculated based on the ID results a nonlinear gradient. So the main purpose of that you can see here. So we want to achieve an equal illusion of all peptides across the whole separation space. If you look at the black bars, you can see there is a drop in IDs. The retention time bin with a linear gradient at the end, and this is more even using a nonlinear gradient. Another point why you want to do that is we look at the full width half max distribution of the peptides eluting. You can see that with a linear gradient, there are some differences among the separation. So with later retention time, the peak actually is broadening. And if you look in the nonlinear gradient, we can actually achieve an almost the peak full width half max stays the same across the whole separation space, which is very crucial for the optimization of the DIA method that will continue on later. So we did the same experiment for the 4 and 6 hour gradient, and for the 8 hour gradients, we derived the gradient from the 6 hour gradient. So that are the first initial data that we acquired using a HeLa sample. So we're actually quite happy with the results. So we got even already in 2 hours, we were able to identify about 100,000 precursors. And most impressive was actually that the peak capacity increased in an almost linear way from the 2 to the 8 hour gradient. And then we compared it also to an in-house pack column. There you could see that actually the full width half max is very similar for 2 hour gradients and also the peak capacities. But this longer gradients, actually you can see that the peak width of the micropack column increased less than compared to the in-house pack column. So then we also looked again into how many identifications per peak capacity were achieved. We could see actually that this is also almost stable line, with the exception of the 8 hour time point. But for that, we know that we didn't really optimize the DIA method for the 8 hour gradient, so therefore it's a suboptimal method. But these initial results showed us already that the micropack column provides us with very high peak capacities for long gradients. With that, we started off to optimize the micropack DIA workflow, starting with the peptide loading on column. The next step will be about the optimization of the DIA method. And then I will briefly say how we generated an optimized library. The readout, how we evaluate the success of this, was to increase the number of the identified precursors, peptides, and proteins using a HeLa sample. And with optimized method, we also looked into the quantitative precision using replicate runs. So our usual loading is around 2 micrograms of peptides. This is a summary where we try to double the loading from 2 to 4, and for the long gradients also from 4 to 6 microgram loading. What you could see here is that we get roughly an increase of 4 to 6% using using an increased loading from 2 microgram to 4 microgram, but then further increase to 6 microgram actually didn't lead to more identification. Therefore, we concluded that 4 microgram loading is optimal to maximize the identifications. So the DIA method strongly depends on the peak width of the chromatography, which basically determines the data points per peak for the quantification method. So for that, the cycle time is crucial, and we looked actually into it in this publication from Buda et al. And there we figured out that using using a readout of peptide identification with a CV below 20%, that actually 8 data points per peak at BIO for a DIA method. Within this application, there was a slightly different definition of the data points per peak, therefore translate actually to 5 data points per peak in a spectronaut experiment. Therefore, for our experiment, we try to aim for 5 data points per peak. The cycle time for an Orbitrap analyzer basically depends on the scan resolution, as well as the number of scans or DIA windows. So, for example, for a 2-second cycle time, it's possible to either acquire 60 scans at 15K, 30 scans at 30K resolution, or 15 scans at 60K resolution. So in terms of selectivity, with the most number of windows, the 15K, the selectivity is the highest. So the MS2 spectra is the least complex. But during the acquisition of the scan, the mass spectrometer is not idle. It starts to collect the ions for the next scan. Therefore, if they have a longer scan, there's more time to collect enough ions. So therefore, theoretically, a higher scan resolution can lead to higher injection times and therefore higher sensitivity. For shorter gradients, around 2 hours and less, we usually have 30K resolution. Turned out to be ideal for a number of IDs. But for longer gradients, we don't really know what's the perfect, therefore it needs to be determined experimentally. So for that, you can see the results here. So we tried 2 different methods, one 30K and one 60K method, where we have the 30K method 60 to 75 windows, and 30 to 36 windows is the 60K method. And basically for all gradients, method using 30K resolution lead to most identification. For the 6-hour gradient, there's not a big difference, but for the 4 and 8 hours, it was clearly beneficial to use the 30K method. And as a readout for the data points per peak, actually turned out that for the 4-hour gradient, we achieved the goal pretty well to get 5 data points per peak. For the 6-hour methods, we are very close to it. You just see a little bit shift of the box plot to higher data points per peak, so there's a little bit room for improvement. For the 8-hour method, we still have also time to use more DIA windows. For the future, we actually decided to use the 6-hour gradient because it seemed to be the best compromise of IDs and sample throughput. As I mentioned already in the introduction, for the targeted analysis of the DIA data, a library is required containing peptide sequences, fragment ion information, as well as retention time, or better to say normalized retention time, or IRTs. This library is typically acquired on using high pH reverse phase fractionation. And this is quite obvious that the library size is very important, so you need to cover as many proteins and peptides as possible in the library to identify them in the DIA run. Also, the retention time actually is pretty crucial. So this is something you can see here in the upper graph. We analyzed the same HeLa sample using a library that was generated on a different LC-MS setup. So medium extraction width in which the software starts to look for the peaks was around 33 minutes in a 6-hour gradient, which translates to 18% of the gradient. And analyzing the same sample with a project-specific HeLa library on the same setup actually reduced this window to 4% of the gradient. As it is not so easy to compare identification between different libraries, they can also differ which fragment ions are in there and so on. We actually did an in silico experiment, basically adding noise to the IRT, made them artificially more imprecise. And the results you can see here. The 0 is basically the high precision IRTs without any noise added. And the more noise is actually added to the retention time, the less peptides get identified. So that actually leads to the conclusion that having the library or made the library generation on the same LC setup actually improves the identification results. So with that, we wanted to push it to the limits. For that, we started off again with a HeLa sample, used the 6-hour gradient, loaded 4 micrograms of peptides. We further optimized the DIA method using 90 windows and 30K resolution. We acquired as a project-specific HeLa library using 15 high pH reverse phase fractions, and the library comprised more than 300,000 precursors and slightly more than 10,000 protein groups. We have actually generated a very deep library, and we did triplicate injections to access the quantitative precision. That's an overview of the results. So we're actually able to identify 9,123 proteins in average per run. This all is filtered by a 1% FDR on protein level. In total, in the experiment with the 3 replicates, we identified 9,173 proteins. And the IDs between the ones were reproducible. So the run with the least IDs was 9,121, and with the highest IDs, 9,124. We were able to cover more than 4 orders of magnitude. Then we also calculated the CV based on the triplicate injection. So the median CV across the whole dynamic range was 6%. But we were also interested how the CV behaves in different abundance regions. Therefore, we just split the dynamic range into 3 parts using the high abundant, medium abundant, and lowly abundant proteins. And there we could see that actually even the low abundant protein range, we still achieved CVs in the range of 10%, and this drops down to 4% for the highly abundant proteins. So this results actually among the highest ever reported protein IDs within a single-shot analysis using a 1% protein FDR. So to give you a better impression of the data reproducibility, you can see here a heat map representation of the 3 runs. They basically look the same with only very minor differences. And also if you look in the number of quantified proteins with a CV below 20%, we were able to identify 8,000 proteins with it. And if you look at the dataset completeness, you had more than 9,000 proteins quantified in all 3 runs, resulting into a dataset completeness of higher than 99%. So in summary, we actually achieved a very highly reproducible and precise quantification on a whole proteome scale. So to give you a little bit an impression of what is possible with this kind of data set, we did some enrichment analysis of the highly, medium, and lowly abundant proteins. Here's the overview about the highly abundant proteins. You can cover, for example, DNA replication proteins and also the oxidative phosphorylation chain. In star, always mark the proteins that were quantified. We actually were able to achieve a high coverage of major metabolic pathways and cell growth-related pathways. For median abundant proteomes, you can see a representation of pancreatic cancer, which sounds a little bit odd in the first moment to look into this pathway for HeLa cell line, but it's basically just a representation that we can achieve a good coverage also of disease-related pathways. For the lowly abundant proteins, we, for example, saw some signaling pathways here, a picture of the toll-like receptor signaling pathways. So that led to the conclusion that we can also get a good coverage of signaling and regulatory pathways, including some transcription factors, which are usually very lowly abundant and therefore hard to quantify. To conclude, so DIA combines sensitivity, reproducibility, precision, scalability for proteomic experiments. The micropack allows high peak capacity for long gradients, longer than 2 hours, with very high reproducibility. We achieved CVs around 6% and the 99% data set completeness. We have a very robust separation. We basically looked at it and how reproducible or how big the XIC extraction window is. And for these ones, we had a width from 2%, and for our in-house pack columns, usually are around 4 to 5%. So in summary, the micropack and DIA enables a single-shot comprehensive proteome analysis covering more than 9,000 proteins in 6 hours with an FDR of 1%. With that, I would like to thank the whole R&D team of Biognosys, especially Roland Bruderer, who acquired the last sample set, the ELA record runs. And from Pharmafluidics, I would like to thank especially Jeff and Paul, who came to our lab to install the first micropack that we had. And with that, I would like to hand over to Laura again.
Thank you very much, Jan. I now have 2 more polling questions for everyone in the audience. And once again, you can click directly on your screen to enter your answers. So this next question is up on your screen. And as you can see, it says, are you considering data-independent acquisition, or DIA, methods? I'm using DIA already. I'm interested in DIA in the near future. Or I'm not interested and I'm happy with my current workflow or workflows. So once again, you can click directly on your screen to enter your answer. And that question again is, are you considering data-independent acquisition methods? And the possible answers are, I'm using DIA already, I am interested in DIA in the near future, or I'm not interested and I'm happy with my current workflow. Okay, let's bring up that result. We've got kind of a wide range of answers there, with almost evenly split between people who are using DIA or want to use it in the future. Fantastic. Okay, we have just one last question for all of you in the audience. And that question is now on your screen, and it says, are you considering a data-independent acquisition method in combination with the micropillar array column? And the answer possibilities are, I'm using it already, or I'm interested in trying this combination in the near future, or I'm not interested and I'm happy with my current workflow. So once again, click directly on your screen to answer. And that question one last time is, are you considering a data-independent acquisition method in combination with the micropillar array column? I'm using it already, I'm interested in trying this combination in the near future, or I'm not interested and I'm happy with my current workflows. All right, let's pull those results. So the vast majority, about 83%, said that they're interested in trying the combination in the near future. Excellent. All right, so I'd like to pass it off to Jeff for a few closing remarks.
So it seems a lot of people are interested in using our columns in combination with data-independent acquisition methods. So if you are interested in one of our products, you can find all information and application notes on our website. We currently have 2 types of columns on the shelf, a 50 centimeter and a 200 centimeter long column, and each column has been functionalized with C18 and is end capped. So with a column volume of 3 microliter and a flow rate range of 0.1 to 2 microliter per minute, the 50 centimeter micropack column is ideally suited to perform this more routine analysis where increased throughput is needed. And for those who are aiming to this really comprehensive proteome coverage, the 200 centimeter long column with a volume of 9 microliter and a flow rate range of 0.1 to 1 microliter per minute is the column you should use. To conclude, we also distribute a micropack column easy spray emitter that can be ordered from our website. Thank you again for your attention, and we are happy to try and answer all of your questions. Back to you, Laura.
Thank you very much, Jeff. It is indeed time for the question and answer period with the audience. As a reminder, if you would like to submit a question, you can do that by typing it in the Q&A box, which you can find on the right-hand side of the window, of your window. All right, so let's get started. So here's our first question. What is the typical number of injections on a micropack column that can be done until the column needs to be replaced?
So I think we have already done some experiments on the micropack column regarding longevity. So we operated the column during 6 months without any loss of column performance. So it was an experiment where we continuously injected HeLa samples with in between some reference material to check the retention, and the column survived these 6 months or 2,500 total runs. So for the time being, we do not really know what the limit of this column is. I do need to say with it that it is a HeLa sample, so it's a quite clean sample. So it will also depend on what sample type or what sample prep you're using for your samples.
Excellent. Here's another question. Have you done any applications of the micropillar array columns using non-proteomics or typical LC conditions for pharmaceutical analysis, such as an impurity method using a flow rate of 0.5 milliliters per minute?
We have already done a lot of other type of separations. I think in the field of metabolomics, or in the field of peptide mapping, and also for the analysis of antibodies, of course, where we did peptide mapping of just a single protein, an antibody. And these application notes are also available on the website. But of course, you can also use this column, the same column that we use for proteomics, for the separation of lipidomes or metabolomes.
Excellent, thank you. Here's another question. This person says, maybe I missed it, but what is the carryover of the column, and what washing procedure have you used for a 6-hour gradient?
So maybe I should answer this one. I'm not sure. Jeff, what do you think?
Yes, you can answer, because it's regarding the 6-hour gradient method.
Yeah, at this point, actually, we didn't assess the carryover. I would just assume, based on the low CV across the replicates, that is actually rather low. So it doesn't look like there's anything accumulating on the column. At this point, we just used 15 minutes for step at the end, but we didn't really look into whether it's too long or too short at this point.
All right, here's another question. We use a trap elute setup with our nano LC column. Is a trap column unnecessary with the MicroPack 200 column, 200 centimeter column, I think that means?
So it of course depends. I think in this configuration, all the experiments were performed using a direct injection. So, but this puts some kind of a constraint on your sample concentration. If you are working with very low concentrated samples, then of course trapping would be beneficial. But at the time being, we do not have our own trapping columns yet. So we are developing them, and they will be available soon. And this is actually important because if you're combining a trapping column with an analytical column, the stationary phases should be adapted to each other. Because if you would have a trapping column that has more attention than the analytical column, the peptides will elute broader or won't have enough retention on the analytical column. So that's why it's crucial for us to also provide trapping columns, but they will be available soon also.
Thank you. Jeff, just a quick note, your audio is not quite as clear as it was at the beginning of the web seminar, so I don't know if there's an adjustment that you can make.
Yeah.
So here's another question. Will further separation phases, apart from C18, become available in the near future?
So at the time being, we're doing research on this topic because we already got a lot of questions regarding this, and we're looking into different chemistries such as Hillic, but at the time being, we do not have them for sale yet. So there are Hillic type of chemistries we are working on, but we're also looking towards shorter chain reversed phase chemistries such as C8 or C4 to do more intact protein work on the columns.
Right. Your audio is better. Thank you. We have a follow-up question on an earlier question. It says, the question on the number of injections refers to practical usage by Dr. Muntel.
Sorry, can you please repeat it? Not sure whether I got it.
Sure. Because the original question was, what is the typical number of injections that you can do on the MicroPack column before the column needs to be replaced? And so now the person is confirming that they want to know from your direct experience of using it, what your experience has been in terms of how often you have to replace the column.
Okay, so far we didn't replace the column at all. So all of the injections that I showed today were acquired on the same column. So I cannot really answer on it, so we just have it on for maybe a week at once. Then you have to change again to a regular setup because we just wanted to evaluate at this point. But so far, we just ran everything on the same column.
Thank you. Here's a new question. Is the use of nonlinear gradients straightforward and compatible with the use of IRT standards? Or does the RT calculation have to be modified?
So actually the IRT calibration, or we now call it high precision IRT, so in the software itself there are now several thousand calibration peptides in it, so actually the software is actually designed to deal with nonlinear gradients. Because we also use them routinely anyway, and therefore it's optimized for that.
How easily can a DIA method be implemented on my system?
So it's actually rather easy. So we also have some pre-prepared methods that we can hand out to customers. So basically, you give us which full width half max you expect from your system, and then we can provide you with a method for it. There is always room for a little bit more improvement, but also as you can see maybe in my presentation, usually it's around 10 to 20% that can optimize on top of that. So usually the DIA method works quite well from the beginning.
Is there any chance of micropack columns with an even higher flow rate or higher flow rates to further enhance sample throughput?
So I think it's regarding the 50 centimeter column. So this has a maximum flow rate of 2 microliter per minute, which is already quite high. But we are, as a matter of fact, also looking into developing columns for a higher flow rate range. So, but this is also something that we are still doing R&D towards, and that will soon, I think, somewhere next year, will become available on the market. But for the time being, our 50 centimeter column with a flow rate of 2 microliter per minute is the highest throughput that we can offer with our system.
Very good. How often do you change the spray emitter tip?
I think perhaps that's something for Jan. Yeah.
I don't know. You can also answer, Jeff, I think.
No. I think it's better for you because you have. I think it's regarding your setup, perhaps, because you have some practical experience with it.
Yeah, I think at the moment we change a little bit more frequently with our regular setup, but it's still maybe once per week maximum in that range. I think there's still some room for improvement there, but I don't. But we still want to change also, check out the other ionization method like the liquid junction injection, which we haven't tried yet.
No.
How is the stationary phase supported by the pillars?
So actually the pillars are first defined into silicon by etching, and then afterwards the pillars are made superficially porous by anodization. So the actual pillar is made porous, so the pores are inside the pillar surface. And then afterwards we attach chemical modification, for instance C18, to this inside the pores. I think this is more or less the answer for the question.
Fantastic. Well, with that, I think we should wrap up. Jeff and Jan, thank you very much for your presentations and for answering all those questions today.
Thank you very much.
No, thanks. No, thank you.
I also want to thank everyone in the audience for attending and participating in today's event. And I'd like to thank our sponsors, Pharmafluidics and Biognosys AG, for making today's webcast possible. At the end of today's webcast, a brief survey will appear on your screen. We hope that you'll take a moment to complete the survey. You will also later receive an email from LCGC alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.